Hello dear students. In today's class we are going to discuss, as you can see, different proteins that are part of the extracellular matrix. Okay, so some parts of the extracellular matrix have already been discussed in previous classes. So today we will specifically discuss some of the proteins. In fact, most importantly, excepting one protein that has been discussed in detail in previous classes. So at first, let us just recapitulate. This has been probably discussed previously. What are the different structures or components of the extracellular matrix? The extracellular matrix basically consists of some proteins and another very important thing, that is proteoglycans. Proteoglycans is a glycoconjugate where carbohydrate with protein, they form the proteoglycan. There are two types of glycoconjugates, proteoglycans and glycoproteins. As you know, it has been discussed in the carbohydrate chemistry classes. So these proteoglycans are a specific type of glycoconjugate where there are some proteins, some central core proteins, and then the glycan or carbohydrate part is mainly GAG or glycosaminoglycan. So that part also has been discussed previously in your classes. So now among the protein parts, if we come to that, there are two groups of proteins mainly. One is structural protein and then there is specialized proteins. Among the structural proteins, the most important three are collagen, elastin and fibrillin. Whereas among the specialized proteins, there are fibronectin and laminin. There are some other minor proteins here and there. We will name some of them also while we discuss. But mainly these are the important proteins. So among them, again, as you can see, the collagen has been discussed in quite a bit of detail in previous classes. So among all the components, proteoglycans previously discussed, and among the proteins, collagen previously discussed. So now when we say that we will discuss proteins of extracellular matrix, so we will discuss other proteins that are, obviously collagen is most important among them, but other important proteins that are there, namely elastin, fibrillin, fibronectin, laminin, etc. So we will discuss all of them one by one. Okay. So at first, let us come to the elastin. So elastin also is somewhat similar to collagen, but there are a lot of differences also. So what is the major purpose of elastin? As its name suggests, it provides extensibility and elastic property, the elastic recoil property wherever it is present. So in the tissues where it is present, it provides an elasticity kind of characteristics. So from that you can guess what are the tissues where it should be present. So it is present in the lung, especially in the alveolar wall, etc. Then in the arterial blood vessels, in the elastic ligaments, some of the cartilages, etc. So these are the spaces, these are the tissues where, for providing the elastic property, elasticity property and extensibility, this elastin protein is present. So at first let me just tell you also that among all these proteins, I will discuss very briefly about only the major interesting and important points to you, because a lot can be discussed about each of these proteins. But among the components, the proteoglycans and GAG and the collagen, these are the two things that you need to know in a lot of detail. The other things you need to know only about the basic concepts about these proteins. So I will not go into too much detail of any of these proteins. I will just discuss shortly little bit the basic points. So these are the tissues elastin is present. So next, one thing which is common to elastin with collagen is, here also you will find that the prolines are post-translationally modified to hydroxyproline, just like it occurs in the collagen, and by the same enzyme, that is the prolyl hydroxylase. But one thing does not happen here, that is lysyl residues to hydroxylysyl, that is not occurring in elastin, that is only in collagen, but in prolyl to hydroxyprolyl, that is occurring in elastin. Okay. Next, another thing that is common to elastin and collagen is the oxidation of lysyl residues by the lysyl oxidase, that you also know. And in collagen, that lysyl residues are oxidized, that forms the corresponding aldehydes, and which help in the covalent cross-linking of the collagen during their maturation. But here the lysyl residues are again oxidized, produce the aldehydes same as the collagen, but here the cross-linking is different. Here the cross-linking takes help of another protein. I told you that I'll name some other proteins whenever they become relevant to the discussion. So here another protein, which is desmosine, helps in the cross-linking of the elastin polypeptides through their oxidized lysyl residues, and they form a special type of cross-link which is known as tetrafunctional cross-link, where 4 different polypeptides they cross-link together. Okay. And in extracellular matrix, here in the elastin, the collagen, you know, the coil has a very fixed property. How many amino acids per turn, what are the amino acid sequences, the repeating amino acid sequences, everything that has been discussed. But in the elastin, there is no such fixed repeatable type of thing. Here they form a random kind of coil, so they can form coils of different proportions. So what can happen, that coil can actually stretch and again recoil. So since it is a random coil, not a fixed very stable coil, therefore it can stretch and recoil, and that is the reason that elastin can provide with the elastic extensibility kind of property in the extracellular matrix. Now, other than elastin, the next protein that we discuss is fibrillin. This fibrillin is a very large glycoprotein. The molecular weight is about 350 kilodalton. You may forget that. You need not remember it, but if you can, then very good. The purpose of fibrillin is to form microfibrils. Some of the microfibrils that are present in the extracellular matrix, the major component of that microfibrils is fibrillin. This protein is the major component of that. Now, what is the function of these microfibrils? So fibrillin will form the microfibrils, and these microfibrils will then form a scaffold. Scaffold, you know, scaffold means it is the base kind of thing on which the elastin will then be deposited on the extracellular matrix. So the function of the microfibrils is to form the scaffold, a net kind of thing on which the network or meshwork on which the elastin will then be deposited in the extracellular matrix. Now, the fibrillin, as you see, is a very important protein in the extracellular matrix of certain tissues, mainly where the elastin also has to deposit there only. So if there is a genetic defect of fibrillin, then one very important disease, Marfan syndrome, takes place. So what is the cause? It is a genetic disease, obviously. The cause is mutation of fibrillin 1 gene. There are different other types of fibrillin genes, but we will not go into that. So fibrillin 1 gene, which is present in chromosome 15, if that is mutated, then a defective fibrillin protein will be formed. And what are the clinical features? So number one, very importantly, it's very important exam wise, it is asked often, very important MCQ question also, that is dislocation of lens. One of the characteristic features of Marfan syndrome is dislocation of lens, which is known as ectopia lentis. In your anatomy classes, probably it has been discussed or will be discussed later, that the lens of our eyes are held at position by certain ligaments, and there you have the elastin and fibrillin. So if that is defective, then the lens cannot be held in its place properly, and as a result there is dislocation of lens. We will not go into much detail of it. You will know it in more detail when probably you go to the ophthalmology classes, but just know it from now that ectopia lentis is one of the characteristic features of Marfan syndrome. The next thing, obviously, the skeletal tissue, there the like ligaments and tendons and joints we have already discussed, there the elastin and fibrillin If fibrillin is defective, then elastin will also not be deposited well. So if fibrillin is defective, there are certain skeletal defects. Very interesting. What happens? The patient have some tall, long extremities and digits. So the person will be very tall. The arms and the legs will be very long, the long extremities, and digits also will be long, which is known as arachnodactyly. And the joints will be unusually hyperextensible. They are very, very flexible. So you can just extend them in many weird ways. So these are some of the features of the skeletal defects of Marfan syndrome. Also, as you can see that, as we have discussed, that arterial wall, etc., there is also presence of fibrillin and elastin. So there will be cardiovascular defect. Sorry, it has been written cardiovascular. There will be a C, cardiovascular defect. Okay. So what happens? There will be weakness of the aortic media. So the aortal wall of the aorta and major arteries, there should be this elastic property. So this property is somewhat lost, and which leads to, in long term, dilation of the ascending aorta, and which may sometimes lead to rupture of the aorta, which may be quite life-threatening. So these are the features of Marfan syndrome. So you can see one patient of Marfan syndrome. You can see this is unusually tall person, but you can see that the arms and legs, specifically in comparison to the torso of the person, you can see this is the torso. The torso is so small torso, and in comparison, the legs are so tall, so long, and the arms are also very, very long. So this is a typical feature of Marfan syndrome. And you can see the digits are also very, very long, unusually long, and this is so long that it can encircle the entire wrist very easily. Okay, so these are the features of Marfan syndrome. One of the typical thing that you can find in Marfan syndrome, that this length, if you stretch all the arms, both the arms, then this length will be more than the height of the person. That is another typical feature that you can find in the Marfan syndrome. Okay, this is what I was telling. This length is more than the height of the person. So the person with Marfan syndrome is tall and thin and has an arm span. This is known as arm span, which is extending, which exceeds the height of the individual. So these are some of the very interesting features of the Marfan syndrome. One of the very famous person from the history, that is, as you can see, you all know him, that is Abraham Lincoln. He probably had Marfan syndrome. We are not sure, but many of his external appearance are very, very suggestive of a probability of Marfan syndrome, but we are not sure. But it is often said that probably he had this. So with this, we finish the discussion of the first two, that is the elastin and the fibrillin. Now we come to the next, that is fibronectin. What is fibronectin? It is again a glycoprotein, obviously present in extracellular matrix. Now fibronectin has two identical subunits, two polypeptides which are exactly same, identical, and they are joined with each other by a disulfide bond. And what else does it do? It binds to the cell membrane. It is present in the extracellular matrix, and it binds to the membrane, cell membrane, via a protein which is a membrane receptor protein known as integrin receptor. And this integrin receptor in itself is a heterodimer of alpha and beta, two polypeptides. Okay, so you can see this is the cell, this is our cell, this is our cell, this is the cell membrane, this is my cell membrane, this is the extracellular matrix. At the cell membrane here we find this is the membrane receptor, that is the integrin receptor. It is a heterodimer, two subunits, one is alpha, one is beta. So A and B actually are alpha and beta subunits, and to this receptor this ECM protein, extracellular matrix protein, fibronectin, binds. And these are the two identical subunits that are joined by a disulfide bond. We will see it better on a next picture. Okay, so some other proteins, extracellular matrix proteins like collagen, like laminin, they also bind to this integrin receptor, but that's beside the point. So this you can see that about the fibronectin. Now, what does it do by binding to the membrane receptor? The thing is that by that way it is linking the proteins of the interior of the cell membrane from the proteins and structures from the extracellular matrix. Okay, so how does it happen? Let us see the diagram. So say this is our cell membrane. This is our cell membrane. This is the inside of the cell, cytosol, and this is the outside of the cell, that is the extracellular matrix. Now what happens? This is our fibronectin protein. As I have told you that there are two identical subunits, subunit 1, subunit 2, and they are joined by disulfide bonds. So this is the homodimer of the fibronectin protein. Now it is present in the extracellular matrix, as you can see, and then it is binding to the integrin receptor that is present in the plasma membrane, having two subunits, alpha and beta. A and B means alpha and beta. Now this integrin receptor in turn is binding to some of the important proteins that are present in the cytosol. Some examples are tallin, vinculin, capping proteins, alpha-actin, etc. If you can remember one or two names, that will be fantastic. Tallin, vinculin, etc. And these proteins then bind with some of the cytosolic proteins which are involved in the cell movement, cell motility, etc., like actin. And here the fibronectin is also binding to extracellular matrix proteins like collagen, sometimes with some other proteins also. We'll not go into much detail, but you can see throughout this structure formation, mainly by the integrin of the cell membrane and fibronectin in the extracellular matrix, they too help in the linking a series of proteins so that there is a communication channel that is formed from the outside of the cell, extracellular matrix, and the inside of the cell, that is these proteins. So these entire things finally help in communication and motility and mobility of the cell on different situations and reacting to different conditions and stimuli, etc. So that is a complex thing. We'll not go into that. But this much you can see that this extracellular matrix protein is very important in forming a communication between some of the proteins of the extracellular matrix and the cytosol. Okay. Next, last protein that we'll discuss is laminin. So about laminin, we'll come to the diagram later. So about laminin, it forms the basal lamina. So laminin forms a very specific structure in the extracellular matrix outside the cell, that is basal lamina. Often we call it basement membrane, basal lamina. Okay. This has three polypeptide chains. In our diagram, this is the laminin. This green one is the laminin. You can see alpha, beta, and gamma. Three chains. Each of these are laminins. Each of these are laminins. They have alpha, beta, gamma, three polypeptide chains. Okay. Now they form a network. There is a network formed by the laminin proteins, as you can see, a network of these laminin proteins. This network then binds to network formed by another ECM protein which is present in the basement membrane, that is a special type of collagen, you know, that is collagen 4. Collagen 4 which is present in the basal lamina or basement membrane. This network, collagen 4 network, this one, this blue one is the collagen, type 4 collagen. Here you can see this one. This collagen network is bound with the network of the laminin. And there are other proteins which help in this binding. What are they? They are enactin and prolactin. This small triangle is enactin and this one is prolactin. These ones, these purple elongated ones are prolactin, as it is written here also. So the network of laminin bind to the network of the collagen 4, and these bindings are helped by some other proteins. I told you many other proteins will come, but the major important ones we are discussing. So enactin and prolactin, they help in this binding. Okay. And then on the other side, this is the binding on the ECM side. Then on the cell membrane side, these laminins again bind to the integrin receptors. The same integrin receptor, similar thing that we discussed previously. Okay, so they again bind to the integrin receptors. So through these things, you can again see there is an interaction, there is a communication, there is a connection made from the cell membrane of the cells and the basal lamina. So that again is functionally very, very important. And this entire structure, we call it the basal lamina. This is specifically present in the extracellular matrix of some of the epithelial cells. This becomes very, very important in the basement membrane, specifically of the glomerular cells. In glomerulus, this basal lamina is very, very important, because on this basal lamina, there are on two sides, there are cells of the blood vessels, on the other side the cells of the nephron. So through this, from the blood to the nephron, the filtration occurs through this, and this helps in the formation of the sieve for certain things. And if that is defective, certain diseases occur. Will not again go into much detail of that, but you can see this is very, very important structure in the extracellular matrix. Okay, so these are the proteins that we know that are important in the extracellular matrix. Collagen, already you know, you have to know every detail about it. It's very, very important. But some other proteins are also quite important, like elastin, fibrillin, fibronectin and laminin. These four you need to know little bit about each of them, little bit about the Marfan syndrome that is occurring due to defect of fibrillin 1, and all these overall concepts. And if you want to read or if you want to study more detail about these topics, I'll suggest you go through the chapter of extracellular matrix in Harper. This is one chapter you can study from Harper. It is in no other book. It is so well written and so much information is there as it is in the Harper. So if you want to read more detail about this topic, please go through Harper. And for those who think just the bare minimum you need to know, then the little bit that I discussed, you have to know this much at least. And then for further study, please go through Harper. Okay. Thank you very much.